Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So in last class we learned about the different types of the X chromosome inactivation or the different methods by which X chromosome can be inactivated. Okay, so today we are going to learn about the stages of X chromosome inactivation. Okay, so in last class we learned that the placental mammals, okay, they show random X chromosome inactivation in the embryonic stem cells, okay, but the imprinted X chromosome inactivation in the extra embryonic tissue, the trophoectoderm, that is going to develop into placenta in future, that shows the imprinted X chromosome inactivation, but the embryonic stem cell in the placental mammals, they show the random X chromosome inactivation, while the marsupials, the primitive mammals, they also show the imprinted X chromosome inactivation. So this is what we learned in the last class. Today we are going to take a look at the stages of X chromosome inactivation. So we are going to learn in detail the step-by-step process by which X chromosome is inactivated. Okay, so the very first step is in the inactivation of the chromosome is counting. Okay, so like first you have to decide whether there is need of the X chromosome inactivation or not. So in the counting we determine sex chromosomes to autosomes ratio. Okay, so we determine this sex chromosome to autosome ratio. For example, in males, they have one X chromosome and a Y chromosome. Okay, so they have one X chromosome on a diploid set of autosomes. So the ratio would be 0.5, the half. Okay, and in the females, they have two X chromosomes. So they have two X on a diploid set of autosomes. So the ratio would be 1. So in the counting, first you determine the X chromosome to autosome ratio, because the determination of this ratio is going to tell us that whether we need the X chromosome inactivation or not. For example, the embryos which have X and Y genetic composition. So the embryo which are going to be male in future, they will have the sex chromosome to autosomes ratio 0.5. So they only have one X chromosome, and that is why there's no need of X chromosome inactivation in there. But if the embryo is female, there you need X chromosome inactivation. So for deciding this, first you need to count the X chromosome. So first you need to determine the X chromosome to autosome ratio, and this counting is done by the some factors which can be either X linked or the factors which can be either autosome linked. Okay, so like by measuring those X-linked or autosome-linked factors, you can count the X chromosome to autosome ratio. So there's not much information available about the autosome-linked factor, but we do know about the X-linked factor. So we are going to discuss about how those factors help us determining the autosome sex chromosome to autosome ratio. How do they do the counting of the X chromosome? Okay. So, for learning about X-linked factors, first let's take a look at the X chromosome inactivation center. So this is XIC, X chromosome inactivation center. So this is X inactivation center, XIC. Okay. Okay, so this is X inactivation center is a region on it. This is the X inactivation center. This is a region on the X chromosome which is going to do the X inactivation. Okay, this is going to play a role in the inactivation of the X chromosome. So this is going to do the heterochromatinization of X chromosome. So this center consists of the Xist non-coding RNA that we discussed in the non-coding RNA part. Okay, it has the antisense transcript of Xist known as T6. Okay, this is antisense of the Xist, and that is why it is written as XIST, and that is why it is named as T6. Okay, so the XIC consists of Xist, T6, and some other factors which are upstream to Xist like FTX, JPX, and the downstream to the Xist like DX, PAS34, Excite, TSX. Okay, so and as well as there's upstream sequence known as RNF12. So all of those are included in the X inactivation center. But all of the factors except the RNF12, all of the regions are non-coding. Okay, so except RNF12, even in the schematic, you can see that the RNF12 is a coding for some protein, while all of them are non-coding. So except for RNF12, all of the regions included in XIC are non-coding. Okay, so let's forget about all this part for now. Let's just focus on the RNF12. Okay, so this RNF12, this codes for a protein. Okay, and this is very important. This is very crucial, and this is going to be helpful in the counting part. Okay, so how? Let's take a look at that. So what happens is that in the females, RNF12 gets inactivated. So RNF12, it is the X-linked gene, right? So when this X-linked gene RNF12, it codes for RNF12 protein. Okay, this RNF12, when it is reached a certain threshold level, this is required for X chromosome inactivation. So what happens is that when RNF12 is produced from X chromosome, RNF12 is produced. Okay, and when the production is enough and RNF12, the amount of RNF12, it crosses certain threshold. When it crosses certain threshold, it activates Xist RNA. Okay, it is going to activate the Xist long non-coding RNA. But for the activation of Xist, it needs to reach certain threshold level. Okay, so when it activates Xist RNA, now this Xist RNA, it is going to do the inactivation of X chromosome. Okay, and this is how RNF12 does the inactivation of X chromosome. But what happens is that for that you need a certain threshold level. Okay, so you need certain amount. Let's say that you need X amount of RNF12 for activation of Xist mRNA. So what happens is that females who have two X chromosomes, they have the double expression compared to that of the males. So the amount of RNF12 would be more in the females compared to the males. So in males, the threshold amount that is required for Xist activation, that is not reached. It is only achieved in the females. Okay, because females have uh, double expression of the X-linked genes, and that is why when in females the threshold level is reached, it activates Xist, and that is why only in females the X chromosome inactivation is seen. Okay, not in males, because in males they do have RNF12. So both females and males, both of them show the RNF12 expression. But in males, it doesn't reach to the certain threshold level, and that is why it is unable to activate the Xist. And now the RNF12 is produced, but it doesn't activate Xist in males, and that is why X chromosome will not be inactivated. But on the contrary, in the females, RNF12 is in sufficient amount, and now it can activate the Xist RNA, and now Xist long non-coding RNA, it is going to inactivate the X chromosome. So this is how RNF12 is the deciding factor of the whether the X chromosome will be inactivated or not, and that will be only inactivated in the case of females. Okay, so like if there's some mutation in males and the RNF12 expression is elevated, it can lead to the X chromosome inactivation. So what is responsible for X chromosome inactivation? RNF12 expression. When it reaches to some threshold, it activates Xist, and now when Xist is activated, it is going to do the X chromosome inactivation. Okay, so this is the flow. This is what happens in the females. Okay, so there's one more thing, that when Xist levels they reach to certain level, level, this leads to the silencing of RNF12. So when Xist is activated in enough amount, it leads to the silencing of RNF12. Now pause the video and think about it. Why this extra step, the silencing of RNF12 is required? Think about it. So what happens is that when you okay, so now you have elevate, so you have the RNF12 and it is going to activate Xist, okay, and this Xist is going to inactivate the X chromosome, but you only need to inactivate one X chromosome. If what if the Xist expression goes up, it goes to that level that it can inactivate both of the X chromosome, right? Because Xist RNA, long non-coding RNA, it acts as a guide. Okay, so it acts as a guide, it covers the X chromosome, and that is how it inactivates the X chromosome. So if the expression is much more than what is desired, it can inactivate both X chromosome. So for avoiding that, once the expression reaches to certain level, we silence the RNF12 expression. So now RNF12 is silenced, so it can't activate Xist further. So it is kind of self-regulation. Okay, so like when RNF12 this reaches to certain level, it activates Xist. Okay, okay, and when Xist reaches to certain level, it silences RNF12. So now no more RNF12 is produced, and that is why only the Xist mRNA which was already produced, that is working. So no more Xist mRNA will be produced, and this, sorry, Xist long non-coding RNA. So the Xist won't be able to inactivate the other X chromosome. So only enough amount which is desired of the Xist long non-coding RNA that will be produced, and for making this happen, we need an extra step of the RNF12 silencing, and that is done only when Xist reaches to a certain level, so that it is enough to inactivate one X chromosome and not the other X chromosome. Okay, so this is how RNF12 is an important factor for determining whether the X chromosome inactivation will be done or not. So if someone asks you a question, whether RNF12 is produced in both males and females, yes, it is produced in both males and females. Then why only females show the X chromosome inactivation? Because for activation of Xist LNC RNA, there is a requirement of certain threshold level of RNF12, and that is reached only in females due to the extra X chromosome copy. Okay, so in males RNF12 is produced, but it doesn't reach to that level so that it can activate Xist. So even if RNF12 is present in males, it doesn't activate Xist because it is not in its enough amount. But in females, it is crossed the threshold level, and now it can activate Xist long non-coding RNA is going to do the inactivation of the X chromosome. So this is it in the counting part. In the next lecture, we will learn about the X chromosome inactivation center in detail. We will also learn about the choice, and we will continue with the further stages of X chromosome inactivation.